এসছে দেখাচ্ছে ঠিক আছে ওকে হ্যাঁ সুব্রত আছে অন্য আইডি দিয়ে আছে ওই ইয়ে দিয়ে অ্যালাউ করেছে আমি দাঁড়া ওর সাথে ইয়ে করছি ইউটিউবে এসছে কিনা দেখ তো আছে অন্য আইডি দিয়ে আছে ওই ইয়ে অ্যালাউ করেছে আমি দাঁড়া ওর সাথে ইয়ে করছি ইউটিউবে এসছে কিনা দেখ তো না না ওখানে একটা আছে অডিও মিক্সার বলে একটা অপশন আছে অডিও মিক্সারের ওই ডেস্কটপ অডিওটাকে অন রাখবো না না ওখানে আমি ইয়েটা উপরের অংশটাকে মানে ক্লোজ করে দিয়েছি হ্যাঁ এইটাকে অফ করার তো অপশন দিচ্ছে না নিচে না ওটাকে কমিয়ে হ্যাঁ সেটাতে ক্রস করে দিচ্ছি কিন্তু নিচেরটা তো মাইক অফসটা দেখাচ্ছে ডেস্কটপ অডিওটা অফ আছে দেখাচ্ছে ওপরের আর মাইক অফসের জায়গাটা তো ডেস্কটপ অডিওটাকে রেখেছি, আর নিচে মাইক অক্সের অপশানটা অফের, এটা অফের জায়গাতে দেখাচ্ছে না। লক ভলিউম করে দেবো, লক ভলিউম, লক।
একটু দেখাও, আমাকে দেখাও ডেস্কটপটাকে। হ্যাঁ হ্যাঁ, ওই মাইক আর অক্স তার নিচের অপশনটা, তা মাইক দেখাচ্ছে না।
ও নিচের, নিচের অপশানটা বন্ধ।
ভলিউমটা কমা। দাক্ষায়ণী আছে?
আশিস জয়েন করেছিস?
হ্যাঁ জয়েন করেছি তো।
আমার তো
এই তো
কোচবিহার
5টায় শুরু হবে। হ্যাঁ বলুন। গুড মর্নিং সবাই, গুড ইভিনিং সবাইকে। আলোকদা কিছু বলছিলেন?
Hello everyone, good evening. Please let me know whether I am audible and my video is working or not.
Yeah, it's fine. It's working.
Okay, thank you very much.
Okay.
Shall we start? Okay. Good evening and welcome to one-day international webinar on current trends of research in biological sciences, Cell to System, organized by Department of Physiology, Acharya Brojendranath Cell College, Kuch Bihar. I Pasang Dukpa, Assistant Professor in the Department of Physiology and your host for the webinar. Now, without further ado, I would like to request Dr. Tridep Chakraborty, Head of the Department of Physiology and convener of the organizing committee of the webinar, to kindly address the online gathering. Over to Dr. Chakraborty, sir.
Thank you, Prashant, for your nice words. A very good evening and heartiest greetings to everybody. Respected Dr. Niloy Rai, Honorable Principal of Acharya Brojendranath Shil College and Patron of the Webinar Organizing Committee, Dr. Arijit Chakraborty, Coordinator of the Internal Quality Assurance Cell, Sri Mrinal Kanti Boshak, Secretary of the Teachers Council, my dear colleagues, teachers from other colleges, universities and institutes, eminent speakers of today's webinar, and all my dear students. On behalf of the Webinar Organizing Committee and Department of Physiology, I feel honored and take the pleasure in welcoming all of you to this inaugural session of the one-day international webinar on current trends of research in biological sciences, Cell to System, organized by our Department of Physiology of Acharya Brojendranath Shil College, which is one of the premier academic institutions located in Kuch Bihar, West Bengal, with its glorious heritage and rich tradition. The significant and enormous achievements being made in different areas of physiology and other disciplines of biological sciences around the globe have necessitated in dissemination of scientific knowledge through organization of conferences, seminars, symposia, etc. However, during this prevailing COVID pandemic situation for the last 1.5 years, together with the lockdown periods, online platforms have proved to be the only choice in continuing teaching learning activities, along with webinars and e-conferences for exchanging scientific knowledge as well. We are very much delighted to have with us today 3 internationally renowned speakers who have made significant contributions in their areas of expertise. Our first speaker of today's webinar is Dr. Shara Behnami of Dartmouth College, New Hampshire, USA. Dr. Behnami will be talking on the power of molecular biology techniques. Our second speaker, Dr. Pallav Sengupta of Masha University, Malaysia, will deliver his lecture on COVID-19 and effects on male reproductive system. And the third speaker of today's webinar is Dr. Upasana Das Adhikari of Reagan Institute of MGH, Harvard, and MIT, USA. She will be talking on host microbiota interactions, modeling of the gastrointestinal tract. In this regard, I must acknowledge our respected principal sir, Dr. Niloy Rai, for his kind support and initiative for organizing such webinars by each and every department of our college for the benefit of students and teachers as well. Sir, we are truly obliged for your inspiration. Further, I sincerely acknowledge my esteemed fellow faculty members, Dr. Tuhin Shubro Banerjee, Organizing Secretary, and Srimati Payal Bonik, Srimati Anindita Chakraborty, and Sri Pasham Sering Dukpa, Joint Organizing Secretaries, for their wholehearted contributions in each and every step for preparation of this webinar. I am so very indebted to them that any of my sincere efforts to acknowledge their contribution will fall short of the actual matter. Moral support from our students is highly appreciable. Last but not the least, the overwhelming response on the number of participants is praiseworthy. This webinar will certainly enrich the participants with current trends of research in biological sciences and generate the scope for interactions with the scientific community. I hope you all will have a pleasant experience and will enjoy the lectures as well. With these words, I express my heartfelt wishes for the success of this international webinar. Thank you very much. Over to you, Prashant sir.
Thank you, sir, for your warm welcome and kind word. Now, I request our patron and honorable principal, sir, Dr. Nilay Ray, to grace us with the inaugural address. Over to you, sir, please.
Thank you, Pasang sir. Good evening, respected head of the department and faculty members, Department of Physiology, ABN Shield College, Kuch Bihar. Respected speakers, Dr. Benami, Dr. Sengupta, and Dr. Das Odhikari. Coordinator IQAC, Secretary Teachers Council, System Administrator, and participants. I'm honored to be in the inaugural session of this international webinar on current trends of research in biological sciences, cell to system. I must congratulate the organizers for their efforts towards arranging such a nice platform to discuss on the issues, the related issues of current trends of research in biological sciences with three important and interesting topics. This century-old college is indebted to Dr. Sarah Behnami, Dartmouth College, USA, Dr. Pallab Sengupta, Masha University, Malaysia, and Dr. Upasana Das Odhikari, Harvard, USA. Thank you, sir and madam, for your gracious presence. Participants from different disciplines and sections from India and abroad will enjoy the deliberations and sharing of views in coming 2 to 3 hours. Wishing a grand success of this webinar. Thank you very much.
I would like to thank our principal, sir, on behalf of the department and organizing committee for his valuable speech and continuous support in organizing this international webinar. Thank you very much, sir, once again. Now, we shall proceed further with the technical session. But before we start with the technical session, I would like to extend our warm welcome to our respected and renowned invited resource persons, Dr. Sarah Behnami, Dr. Pallab Sengupta, and Dr. Upasana Das Adhikari. We are very much honored and privileged to have you all with us today. Thank you for joining us. Now, before we begin with the first lecture, this is for the kind information to all the participants that we shall be having a short interactive session of 10 minutes at the end of each lecture. Any questions you may have, kindly post it in the Google Meet inbox or in the YouTube comment box. We shall refer to them at the end of the lecture. And also to all the participants who are joining us through the Google Meet, you are requested to keep your video off and microphone in mute mode for the entire session of the webinar. Once again, I would like to request all the participants who are joining us through the Google Meet to keep their video off and microphone in mute mode for the entire session of the webinar. Now we shall begin with our first lecture. We have with us Dr. Sarah Behnami, and she will be talking in the topic, the power of molecular techniques. Dr. Sarah Behnami is a cellular and molecular biologist. Dr. Behnami completed her PhD from Orontario Tech University, Canada. She is presently continuing her research as a postdoctoral fellow in Dartmouth College, New Hampshire, USA. She has innumerable published research articles in esteemed journals like Human Reproduction, Chemistry, and so on. Her area of research interest includes different cellular and molecular pathways that contribute to herbicide tolerance, employing a wide variety of methods at the interface of genetics, molecular and cellular biology, proteomics, and biochemistry in vitro and in vivo systems. Dr. Behnami has also been working as teaching assistant at Ontario Tech University since 2014. She was awarded with the prestigious MITAX Research Training Award in 2020. Now with this, I request Dr. Behnami Madam to begin with her lecture. Over to Madam.
Hi, and thank you very much for your kind words and very nice introduction. It's my absolute pleasure to be here today and give a lecture to all the people from around the world. And today I am going to talk about the power of molecular biology techniques, which is one of the very most important aspects of biology research and medical and non-medical research. So with this, I would like to share my screen with you guys. So the power of molecular biology techniques. First, let's see what we're going to talk about here. Molecular biology technique, basically they utilize the DNA, RNA, and enzyme that interact with nucleic acids to understand biology at the molecular level. To just a very brief introduction about what is nucleic acids. Nucleic acids are genetic material of all known organisms. For example, DNA or deoxyribonucleic acid, RNA, ribonucleic acid, which present in some viruses. They also consist of chemically linked sequences of nucleotides. These links are based on the nitrogenous bases and the sugar, pentose 5-carbon sugar, ribose or deoxyribose, and also they contain a phosphate group. The sequence of bases provide the genetic information. What are the key questions here? First, we want to know what techniques are used for isolating and manipulating these fragments of the molecules, for example, DNA, or in the other word, DNA cloning, and how we can amplify the DNA with the technique called PCR, which I will get to it shortly. And also, we can sequence the DNA. What is these techniques, sequencing DNA? And also, introducing DNA into an organism. These are the some of these key questions. Also, what are the new and precise genome engineering techniques? And how have new genome engineering techniques revolutionized reverse genetic studies and provide us with deeper understanding of the gene function and their expression? Let's start with detecting and quantifying DNA, RNA, and protein. There are methods such as blotting that we can detect DNA, RNA, and protein in living organism or in the laboratory tubes. For example, in vitro, which when we do an experiment in the laboratory tubes, Southern blot or PCR polymerase chain reaction, there are methods to detect and quantify DNA. For RNA, you're using Northern blot or reverse transcription polymerase chain reaction. And for protein, the technique is called Western blot. Also, we can detect and quantify DNA, RNA, or protein in the living organism or in vivo. For example, the technique called FISH, fluorescent in situ hybridization, for detecting a DNA or RNA fragment in situ hybridization for RNA, which the difference between the other one, it's just this one doesn't have the fluorescence, and we use the DNA or RNA fragment. And for protein detection, we use immunofluorescence, which as a probe, we use antibody. Let's get into a deeper aspect of these techniques. And I want to draw your attention to the differences in these techniques. For example, in southern blot usually identifies DNA, while northern blot is RNA and western blot protein, as I mentioned in the previous slide. In southern blot, the probe and northern blot, the probe that we are using is usually labeled with the radioactive molecules and nucleotides that are complementary to the nucleic acid of interest. But in western blot, usually we use the chemiluminescence or fluorescence antibody. As you can see in the figures, at the right figure, you can see the result, it's more or less the same. So we will be able to detect the molecules, either it's RNA, DNA, or protein in the same way by using a different molecule as a probe or as a starting material. For the southern and northern blot analysis, usually we, specifically for DNA here, we digest our molecule, the DNA molecule with the restriction enzyme, and we run them on the electrophoresis and then on the agarose gel with the electrophoresis, and it causes the denaturing the single strand DNA, and we will be able to transfer the single strand DNA from gel to a nitrocellulose or nylon membrane that enable us to visualize the molecules and detect them. DNA of the interest is usually located by hybridization with a labeled probe followed by detection. Here you can see a picture of a northern blot, which we use the RNA as a starting material, and we use the probe, radioactive probe, to be able to visualize as well as quantifying the different the RNA in different organisms. You can see here, we will be able easily to visualize them. This is on the nylon membrane, and you can see that in the different organisms, it's all possible. So what is the genetic engineering? Genetic engineering begins with the cloning of the gene. Basically, we're producing a large amount of gene products. When we talk about cloning, when we say cloning, basically it means, in a very simple word, we try to produce a large amount of that specific gene or molecule, either RNA or protein, that we are trying to detect. Gene cloning requires the isolation of, starting with the isolation of DNA fragment corresponding to the gene of interest, splicing the fragment into a cloning vector like a plasmid. What is happening here, it's basically we try to insert that piece of fragment that we are interested in to study into the vector, into the cloning vector, and introduce that vector to the host, that the host cell will be able to amplify and make a large copy number of that for us. I want also to add this, that the molecular genetics followed by the discovery of DNA structure, which is going back in 1953, and in 1970s, researchers discovered the DNA in the tube that they could easily clone it, and then this led to the recombinant DNA technology, which today we call it genetic engineering. I said recombinant DNA. So for generating recombinant DNA molecule, we need a cloning vector which will be able to replicate in the host cell. By inserting a DNA fragment into a cloning vector, we will be able to amplify and produce a large number of that. But we need a complementary ends for both the DNA fragment and the vector. They will be able to assemble and combine to each other. So which we will be able to do that by the restriction enzyme, which I will be talking about it very shortly. Combination of these two DNA fragments, it happens, DNA fragments, I mean the gene of interest, the DNA, and the vector. So this will happen in the presence of that enzyme. It's called DNA ligase, which seals the nick in DNA. And as a result, instead of two linearized DNA fragments, we will have symmetrical and usually consist of 4 to 8 base pairs. The symmetry occurs at the midpoint formed on opposite DNA end by inverted DNA sequence, or known as palindromes. This question always rises that how the DNA host is not affected by these enzymes. The thing is, the host DNA is protected by base pair modifications, for example, like methylations, which a non-host DNA it's been recognized by the restriction sites and it will be modified, but the host DNA won't be recognized by this enzyme. So this enzyme, I should add also, we get this enzyme from the specific bacteria. For example, the EcoR1 enzyme is from the E. coli, which they synthesized and they use for specific recognition sites. The other technique that we can amplify and we can clone, we can use in the cloning system is polymerase chain reaction, or PCR. This technique is used to make a large amount of DNA sequence, such as other techniques, without the need for cloning. And it's using the gene bank or libraries. So for this technique, it requires a pair of primer, which are a short piece of chemically synthesized DNA. We need an enzyme, polymerase enzyme, or DNA tag polymerase enzyme specifically to synthesize the DNA strand. And also we need exonucleoside triphosphate as a material, and also which calls DNTP, and also we need the DNA template, which usually is our gene of interest, specific fragment of our gene of interest we wish to amplify. PCR process involves in denaturing steps, beginning with the heat that we heated the DNA sample and we cause a denaturation step. So the two strands of DNA will be separated, and in the second step, which is annealing step, the primers with the right temperature, they will be, which is a heat-stable DNA polymerase added, and the primers allow to bind to a single strand DNA, which is created in the previous step by denaturation. So the primers will add it. At this time, in the extension step, with the help of enzyme polymerase and the primers and DNTP, the rest of the template will be synthesized to make, and they make a double-strand DNA. These 3 steps are repeated many times, 2 to 30, sometimes to 40 cycles. And after 20 cycles, a single DNA molecule is modified to 1 million copies. And after 30 cycles, we will have it 1 billion copies. Here, I'll just try to visualize the different step of the PCR. As I said, in the first step, we want to amplify this, the orange section of the gene, which is the gene of the target DNA, to be amplified in our gene of interest. We added the primer and we heat it to 95 degrees. We'll be able to separate the two strands, and then annealing temperature, which is suitable for primer, annealing the primer pieces, which shows in the very small pieces here, and also for the activation of the enzyme. So the two strands are separated. We have single strand. The primers we're adding with the DNTP and enzyme, they start to synthesize a complementary strand. As a result, we will have two DNA fragments, double-strand DNA fragments. Repeats. So from here, the step 2, 3, and 4 for the separation of the two strand, annealing, and extension will be repeated over and over. And then as a result, after 20, 30, 25, or 30 cycle, we will have and I should add here, each of these double-strand DNA act as a template DNA. So they will start separately each of these steps and going over, and as a result, we will have billions of the templates. So there are other ways to clone a gene. For example, making a library, a genomic library, cDNA library, or expression library. To just explain shortly what are each of these are, and then I will get to it in details. For the library, basically is collection of DNA fragments from a single source. These are created for the purpose of isolating specific DNA or genes, which the DNA is part of our gene of interest. For the genomic library, clones of all the DNA from an organism. So this is not only a gene of interest. For the genomic library, we try to clone the entire, all the DNA from an organism. For the cDNA library, which has its own specific purpose, we clone an individual gene or DNA sequence of interest, and the purpose specifically is for the genes that they have a specific functions, and they are translating to proteins specifically. For expression library, clones of gene fragments that can be efficiently expressed as protein in the host cells. For the genomic library, as I said, genomic DNA is cut with a single restriction enzyme and combined with the plasmid vector that has been cut with the same enzyme. Basically, they have the same complementary ends, and this allowed to randomly rejoin by the complementary ends to form a circle. Each new recombinant molecule with the different insert is transformed in a bacterial cell. I have to just remind you, for the genomic library, we were trying to just clone for the entire organism. So we cut these with the one restriction enzyme, different pieces of the gene, different genes, and we have one host vector, plasmid vector, that is cut with the same restriction enzyme, so different gene can recombine into the vector. Then the host cells are selected on nutrient agar containing an antibiotic for selection of the plasmid. What does this mean? Each plasmid, it contains a gene which is an antibiotic resistant gene. So when we say we're selecting it on the nutrient agar containing antibiotic, only those vectors and those cells that containing these antibiotic resistant gene will be able to grow. So we get whatever we are getting at the end, it will be the result of recombination of the DNA into the plasmid. So whatever it doesn't contain and cannot resist to the antibiotic, they won't be able to grow. Then cell multiply and take and make multiple copies of the recombinant plasmid. On the other hand, for cDNA libraries, which is more specific than genomic DNA library, we do not clone all the donor DNA. Instead, focus on protein coding DNA. In this system, mRNA is used as a template to make DNA, because we know the mRNA will translate it to the proteins, so only those DNA that will be able to be function as a protein, work as a functional protein, they will be, it's been used in this library. In this system, oligo-DT is hybridized to poly-A tail, as you can see here, and they anneal to poly-A tail of the mRNA, and the viral reverse transcriptase, which is RNA-dependent DNA polymerase. It's a specific DNA that is translated and that can act with the RNA, which, because we have RNA here, mRNA here, is used to make a single strand DNA copy or cDNA or complementary DNA. From there, the mRNA template, which we don't need it anymore, it will be destroyed with an enzyme, RNase, and the hairpin end of the cDNA now act as a primer to extend and synthesize the complementary strand by the enzyme, which is the DNA polymerase here. So it produces blunt end molecules that can be cloned directly into the vector or modified by addition restriction site on the end on each end, and then we can carry on with the cloning. We talk about the PCR. So PCR, it's not only for it's not being only used for making a large amount of DNA. One of the purpose of the PCR, it's used in cloning, it's used in DNA assembly method. In DNA assembly method, which is in this system actually makes it very easy to modify the sequence of the gene for that specific purpose of our research. DNA assembly, or it's called the Gibson assembly as well, employs three enzymatic activity in a single tube reaction. So it's a 5' and exonuclease, the 3' extension activity of the DNA polymerase, and the DNA ligase activity. These are the three enzymes that is being used for this system. What is happening? First, we use the PCR to amplify and to produce a specific gene of interest that we need to use. The other advantage of this technique is that if we want two different genes that they are in different organism or in different cells or they have a completely different function and they are far apart in the organism, we amplify them and we can actually assemble them next to each other and then in the tube and put them back into the organism. So what is happening here? The 5' exonuclease activity I have to add this here that we use the primers, the usual PCR, we use the primers to amplify these pieces, but these primers also, they have an overlap with the other DNA fragment. So you see here, the fragment A, it amplifies the parts which shows in the orange. It's the overlap with the vector. The red is the overlap with the fragment B, and in fragment B also we have the overlap with fragment A, and also we have the overlap with the other end of the vector. So we amplify these with the PCR, and also we have our vector with the complementary ends, with the overlapping ends, and then now in one single tube with the help of the 3 enzymes, they will be able to assemble to each other, and we can create a very unique DNA assembly that naturally it doesn't exist in the cell. So what's happening? So the 5' exonuclease activity choose back the 5' end sequence and expose the complementary sequence for annealing. Here, the polymerase activity started and fills the gaps on the annealed region, and at the end, the DNA ligase seals the nick and covalently links the DNA fragments. So at the end, we have the assembled DNA, and by transformation and other technique, we will be able to, and the cloning technique, we will be able to amplify and produce a large number of that. Okay, let's talk about the genetic variation. One of the purpose of we are doing all these cloning and gene studies is that there are genetic variations. Most genes have small sequence difference between individuals, which usually occurs every 1,350 base pair on average. These changes, usually it called polymorphism, that these polymorphism can have a very sometimes severe effects on the protein, that's how well the protein works and how the protein even interact with other protein and substrate. So that's very important. If a function of protein is changed in any organism, human or any other organism, can have a very severe damages or can create a new pathway or a new interaction with other proteins. The different gene forms containing polymorphism, usually they call alleles. And these changes, sometimes we call them mutation. And to detect these mutations, there are molecular biology techniques to detect those, which blotting or hybridization method that I explained earlier, that it's one of them. And also we use the sequencing DNA. We compare the mutated and the changes of those nucleotide or those DNA with the original one, or in the other word, we have the mutated organism and the wild-type organism. So we can, by sequencing DNA, we will be able to specify the exact location of the mutation. Also, we can use the restriction enzyme polymorphism. Remember, restriction enzyme, they had a recognition site. So those enzymes, they could recognize only specific nucleotides. If the nucleotides is changed, so the enzyme, it won't be able to recognize and cut that site. So this will be another technique that we can detect the mutation. And also the single nucleotide polymorphism, which is SNP, which we use it for, we can detect that by other methods such as sequencing or restriction enzyme polymorphism. Okay, let's a little bit talk about the sequencing. There are a very, very important methods in recognizing the mutation and the polymorphism happening in the DNA, and I should say many disease, it just causes by one single mutation in the DNA, as I'm sure all of you know, and then the misfunctioning or malfunctioning of a protein caused by these mutations. So sequencing is a very important part of detecting those mutations. So in the next few slides, I'm going to talk about the different method of sequencing. So first, let's talk about the Sanger sequencing. It's a very common method, also known as a chain termination method. It's a method for determining the nucleic sequence of DNA based on the selective incorporation of chain terminating deoxynucleotide by DNA polymorphism during the DNA replication in vitro. So it has a very wide use for, and usually it's used for smaller scale projects, and it needs the validation by next generation sequencing, which is a more complicated and advanced method. The other method is sequencing using the radioactive label. This is a very old technique that works in the same way as a fluorescent label, except that a single DNTP added to the extension mixture. So there are 4 reactions mixture that have to be set for each sequencing. So these fluorescent label, these radioactive label, which is not being very much used these days, but it was a very groundbreaking at the time. So they radioactive label each of the DNTPs, DATP, DTTP, DCTP, and DGTP, and then so when they run them on the gel and when they detect it, they will be able to find out which each nucleotide. So as a result, because we know that based on the sequence and based on the order, we will be able to find out about the sequence from the gel. So, and then we will be able to compare it to the original strand. As I said, this is a very old label, a very old technique, which they don't use the radioactive label these days anymore, and instead they use the fluorescent label. But for the fluorescence label, it's happening the same, more or less the same technique, but in this technique, target DNA or template can be in a plasmid, or they can amplify by the PCR. And DNA primers, usually 15 to 20 base pair, is specific for target the specific region that we are planning to sequence. So target DNA is denatured to a single strand, and the primer is annealed, those fluorescent label primer, and then the DNA polymerase is added along with the mixture of those DNTPs that fluorescently labeled, and they will be mixed, and then as a result, we will be able to, when we run them, as a result, because this is, it's going to be these fluorescents, we don't run them on the gel anymore. The machine will detect the fluorescent, which has a fluorescent scanner, will be able to detect each of the peak that's related to that specific fluorescent dye. This is the output you see from the automated sequencer, that each of these nucleotide, it has a different fluorescent dye. For example, here, the T or A, G or C, they have a difference. So we will be able to, based on each peak and each color, we will be able to detect and have the sequence and able to compare them with the sequence, with the original sequence of the wild type, or non-transgenic or non-mutated DNA that we have. All of these techniques, it's highly recommended to confirm with the next generation sequencing, which is a very high and advanced and very accurate way of these detecting all of these mutations and the sequences. An example of the next generation sequencing is Illumina sequencing, and they will be able to, with this technique, we will be able to sequence the entire genome of an organism. So if I want to just talk about this technique, I should say that the input sample fragments usually fragmented into short length of 100 to 150 base pair. The fragments are ligated to a generic adapter and annealed to a slide using an adapter. Then we use the PCR to amplify each fragment and create a spot with many copies of the same read. They are then separated into a single strand for the sequencing. With this technology, you will be able to, as I said, to sequence about 1 trillion base in 60 hours. And usually it takes a very long time, and we will be able to, sequence the entire genome of the organism. So, in this slide, it shows how exactly it works. The process is repeated by adding 1 nucleotide at a time and imaging in between. So each nucleotide will be added, and then the imaging is happening after adding each nucleotide. So the computer then used to detect the bases at each site in each image, and these are they used to each base is used to construct a sequence. And as a result, we will have by detecting each of them, we will be able to have a final sequence and see what are the sequences exactly. The main advantage, the main thing about this over other sequencing, that's first of all, we can do it in a very large scale. The whole genome will be able, and it's very accurate because after, as I mentioned, after adding each nucleotide, it will be imaging and it will be very accurate. We know exactly what will be the next, what will be exactly the sequence after adding each nucleotide. Okay, these were the techniques that we can find about each mutation or any changes in the sequencing. Going back to engineering genomes, for engineering genomes and even creating these mutations that I mentioned, we can detect them through all these techniques. It's not only happening randomly or for detecting a disease or any specific changes in the DNA or protein. We actually, this gives us this power to create these mutations and to find out about what's going to happen if we creating this mutation and what changes it makes. For example, for creating transgenic plants, it's one of the methods. It's the genomic engineering being used. So we use this genetic engineering or engineering genomes in different organisms. We can basically do in any organism. For example, here is the example for the yeast. The DNA of interest inserted to the yeast chromosome by homologous recombination. The homologous recombination also, it's a technique by two homology fragments will be able to recognize the original gene, and the one that we made any changes or any mutation that we are planning to insert to the original chromosome and insert it to the genome of the organism. These two, number 1 and number 2, it has to be homologous and it should not have any changes. So these two, it will recognize the sites, the exact sites in the genome, and they easily can exchange the newly synthesized the green part, the gene X1, it can be inserted and then replaced by the gene X2. So it will be both ways. We can correct the mutation by this way, or we can create a mutation by this way. Genetic engineering also happening in plants. So we can create transgenic plants generated with the randomly insertion and into the chromosome, that in the plant specifically we use the TI plasmid, which carries a gene of interest and is usually delivered by agrobacterium, and will be able to easily and very efficiently, by just one drop of these tDNA, one drop of our construct on the leaves, we will be able to transfer these genes, these TI plasmid, which contain the tDNA or the randomly modified genes into the plant and create a transgenic plants. Also, genetic engineering happens in animals. The C. elegans is one of the models that is used widely for many disease, for finding about the mutation and curing the disease in different animals. So transgenic worms are generated by injecting a plasmid containing a gene of interest into the plasmid. Again, this is a very simple, by creating, as I explained, by genetic, by cloning, we can create those DNA and those plasmid that they're containing our gene of interest, and the modification, we can easily inject them to the C. elegans, and the C. elegans will take the foreign DNA and replace it into the original gene. Or also, one important aspect of genetic engineering is we will be able to insert even the same gene in some other place other than it's usually located and in the original place. So we will be able to compare the mutated as well as the original and not mutated form of the gene, which usually happens in those models that we need to see that if the gene is homozygous or heterozygous. The last but not the least, definitely, it's the CRISPR-Cas9 genome engineering. This method, which is a very, very highly used these days, and it's actually it's one of the trends and one of the very efficient way of genetic engineering and very accurate. In this method, which I'm sure all of you know, it's we can edit the DNA virtually at any locus. So there won't be any limitation to gene modification. Basically, in any place, any region of the genome, we can make these changes. CRISPR technology actually, it's opened so many doors. and is igniting a revolution across the life sciences. And it very quickly it became a very standard method. So if I want to just give you a very short introduction, because CRISPR-Cas9, we can talk about it hours and hours, which doesn't really fit in this lecture, but I'm going to give you an overview and most important things about this system. The CRISPR-Cas9 system actually compromises a guide RNA or gRNA and a Cas9, basically an enzyme. It's a Cas9 nuclease, which together with the guide RNA form a RNP or ribonucleoprotein complex, and the presence of the specific protospacer motif in the genomic, which usually it has to be very close to the site that we need to modify. So basically, this motif in the genomic DNA, this protospacer is required for the guide RNA to bind to the target sequence, the sequence that we are going to study or making any modification. The Cas9 nuclease then makes a double strand break in DNA, and which is here it shows as a Caesar. So so you can cut it here, and the endogenous repair mechanism triggered by a double strand break may result in gene knockouts, changing in the gene via a frame shift mutation or knocking by adding a DNA sequence in the template. So this technique, it's been used for a variety of purposes, for curing the disease curing the disease to creating a different drug for cancers or other disease. So these were just the highlights and the most important techniques that they were being used in the molecular biology, which enable us to find out about different function and different aspects of the molecular biology. So I hope it was helpful for you and you enjoyed this information. And I'm thanking all of you for being here today and listening to this talk. And now, if you have any question, I'll be more than happy to answer all of your questions.
Thank you, madam, for a wonderful lecture on the various biological tools. This webinar, actually, we are targeting our students. Since because of this pandemic situation, hands-on training is a far-fetched idea right now. So with the help of your lecture, I believe that our students have understand this molecular biology techniques very well. So we are very much obliged and elated for your kind acceptance of being resource person and for this elaborate discussion. Now we shall proceed further with the interactive session. We have with us a couple of questions.
Sure.
So, madam, I kindly request you to address them. The first question we have here is by the Guddu Bhattacharya. Okay. So the question here is that how the annealing temperature and denaturing temperature are determined.
So it's a very good question. So the DNA for the denaturing temperature, it's actually the temperature that the two DNA strand, it caused the two DNA strand just denature and make a single strand. So usually between 95 to 98 degrees,